Well, next is Jonathan Williams from the Baum Lab in the Department of Chemistry and Chemical Biology at Rutgers. Jonathan was mentor to the summer research participants and bootcamp participants who were focusing on the spike, and has also been working on the spike in the Baum Lab. So over to you, John.
Yeah, thank you, Sagar, for that introduction. So yeah, as Sagar said, I'm a postdoctoral associate in the Baum Lab, and since the pandemic has started, we've kind of been interested in looking at the spike protein, and specifically the receptor binding domain of the spike protein, and studying how that interacts with the ACE2 receptor on our own cells, and studying that interaction. So today, let's see if my slide goes ahead here. There we go. So today, I'm going to give kind of a general outline, looking at how the spike protein plays into the virus life cycle, some aspects of the structural biology that are interesting. And so this will be kind of the bulk of the talk at the beginning, hopefully hitting all of the interesting and important points of the spike protein. And then at the end, I'll talk a little bit about, as we've seen with other proteins, the evolution of the spike protein during the pandemic, and the mutations that have cropped up. And some of these you may have heard due to media coverage. So the talks that we've heard, previous to this one, have focused on the non-structural proteins, the NSPs, and now we're moving into the structural region here. Where we talk, I'll be focusing on the spike protein here, which is shown in pink. And the bulk of the structures that have been deposited so far in the PDB have focused on the extra viral, the extra cellular portion of the spike protein. But it is a membrane embedded protein, and it has this stalk that sits within the viral membrane. So to go into a little bit more detail of the spike protein, it is, as I said, a membrane anchored. It's a homotrimeric fusion protein complex, and where each monomer of the protein is roughly 1,273 residues. And so this is quite a large trimeric complex. And as you can see here, it dots the outside of the coronavirus virion. And it's readily apparent why this is known as a spike protein, as it spikes out of the virion. And so it's an important target for study, just from the fundamental aspects of how this mediates the interaction of the coronavirus with our cells, as well as being a point of target for vaccine development and antibody development that targets the spike protein. So within the PDB, there are quite a few structures now deposited on the CoV-2 spike protein, and they kind of cover a lot of different conformational states of the spike protein. So one of the major conformational states that we'll see in the subsequent slides are this kind of change of the receptor binding domain located at the very top of the spike protein here, as it moves from a closed conformation to an open conformation. And this movement of the RBD is really what allows the RBD to be in a binding competent state. And so there are several different forms of the spike protein. The spike protein is quite large and undergoes quite large conformational transitions during its functional life cycle. But before we get into talking a little bit more about the function, I want to kind of talk about the other part of the name that we kind of ignore sometimes, which is the glyco or the glycosylation part of the spike glycoprotein. And so viral glycosylation can play several roles, which include mediating protein folding stability, and they also can facilitate some immune evasion by shielding different parts of the protein from neutralizing antibodies. And so we see here from Watanabe and coworkers in a recent science paper where they use mass spectrometry analysis to determine all of the glycosylation sites on the spike protein. And what they have identified are 22 end-linked glycans per monomer, so 66 end-linked glycans per spike protein. And you can see they dot and cover essentially almost a very large portion of the surface of the spike protein, and they are mapped here in this linear sequence diagram. And so it's important to not forget the glyco part of the spike protein, although at this point we're not going to talk really anymore about that for discussing the functional role of the spike protein. So here I want to talk about the kind of structural biology, the different sequence domains that are part of this class 1 fusion protein. And really these sequence domains kind of help us understand the overall function of the spike protein. So the spike protein in general has two main functions, and they can be kind of described as occurring in two different domains of the spike protein. The first domain is the S1 domain with these 4 or 5 different subdomains. And this S1 domain is responsible for the first function of the spike protein, which is attachment to the host cell. And so those domains that make up the S1 domain are the N-terminal domain. This has, in other coronavirus or other related viruses, has some receptor binding function. So far in SARS-CoV-2, the NTD has not been found to show any sort of receptor binding activity. And then the main receptor binding domain, as its name implies, the RBD, shown here in green at the top of the spike protein. And this is what is responsible for attachment to the ACE2 receptor on human host cells. And then to round out this first S1 domain, we have two subdomains, SD1 and SD2, shown here in purple. It's kind of hard to see, and this cyan color here. And these are kind of linker domains between the RBD to the rest of the spike protein. And so then the second domain, aptly called S2, is where the fusion machinery is located in the spike protein. And so shown here in green, and there's a piece of that shown here on this diagram as well, is the fusion peptide. And so after some cleavage events, which we'll discuss on the next couple slides, this is freed up and then can embed itself in the host membrane, which begins the process of fusing the two membranes together, the virus and the host cell. This is followed by a couple heptad repeat domains, and these are very indicative of the classic type 1 membrane fusion systems. And there are some peptide inhibitors that I believe were developed for previous SARS and MERS viruses that shown some efficacy in inhibiting the membrane fusion activity, although I don't believe I've seen anything yet in that kind of regards to the SARS-CoV-2. And then to round out the S2 domain, we have the transmembrane anchoring domain and the cytoplasmic tail, which sit in the virus itself and anchor the spike protein to the exterior of the virion. So let's discuss the first kind of role of the spike protein in a SARS-CoV-2 infection. So as we saw before, the spike protein decorates the outside of the virus. And at the very tip of that spike protein sits the RBD, and that's what's responsible for interacting with the host cell receptor. In this case, this is the angiotensin 2 receptor, the ACE2 receptor. So in this atomistic picture here on the right, you can see the trimeric spike protein, one of the monomer units highlighted in blue, and then the RBD in the up or open position is able to then interact with the ACE2 highlighted here in magenta. And so it's this initial interaction and the availability of that RBD being in an up position that allows the coronavirus to initially attach to a host cell. And so there's lots of efforts focused in labs around the world, and our lab as well, trying to understand this interaction in better detail, both at the perspective of the conformation of each of the two components, as well as their conformational dynamics, or how the conformation changes with time, and whether we can identify different binding competent states. And of course, all of this is trying to better understand its interaction and potentially then provide information to those who are going to develop therapeutics that may potentially target this interaction for interruption. So during the process of viral entry, the spike protein must be cleaved in a couple places in order, first of all, to allow the fusion machinery that we see kind of contained within the central part of the spike protein in its pre-fusion state to be accessible for fusing the two membranes. And so there are two primary cleavage sites of the spike protein, one occurring between the S1 and S2 domain that again frees up the S2 domain for fusion activity, and then a secondary one that cleaves at the essentially the beginning of the fusion peptide, which then frees it up for attachment to the host cell. And so it has been found for SARS-CoV-2 that a serine protease, a transmembrane bound serine protease TMPRSS2 is necessary for cleavage between the S1 S2 domains. This is known for both, I believe, SARS-CoV-2 and SARS-CoV. And so that cleavage site is kind of conserved between these two SARS coronaviruses as well as other related coronaviruses. What you see here, in addition, in this same binding region or cleavage region, is the addition of a proline, two argines, and an alanine. So this PRRA, these PRRA residues. And so what that does actually is adds in a polybasic cleavage recognition motif between, in addition to the normal cleavage recognition motif, between the S1, S2 subdomains that allows another enzyme, furin, to recognize and cleave between the S1, S2 domains. So instead of just normally having the TMPRSS2, now we have the ability of furin enzyme to come in there and cleave between the two S1 and S2 domains. Now the secondary cleavage site S2 prime that frees up the fusion protein, it's not been conclusively ID'd which enzymes are responsible for cleaving here, although because of the similar nature of the site of, or the sequence of cleavage, it's likely these are furin-like proteases or serine proteases similar to the S1, S2 cleavage site. And so after we have the spike protein cleave now, it can perform its second function, which is fusing the virus itself to the host, creating this fusion pore and allowing all of the RNP material on the interior of the virus to enter the host cell. And so here on the left, we have this illustration that we saw earlier today from David, which is a very nice illustration. And here in a cartoon on the right, we're going to kind of walk through the steps that the fusion process takes. So in its pre-fusion state, we have the spike protein sitting in the virus, and this kind of is reminiscent of that trimeric PDB structures that we observe. That's shown here, letter A. So the spike protein in its pre-fusion state. It comes in, it gets cleaved, it attaches to ACE2, so there's the ACE2 receptor here in the membrane, and the S1 domain is cleaved off. Either it's attached to the ACE2 or it can diffuse away into the cellular environment. And so then after the secondary cleavage that frees up the fusion peptide, then we can have that piece of the spike now embed itself into the host membrane here, shown in B. These are called pre-hairpins, and these cluster together typically. After that, we have some steps where these hairpins start having large conformational changes, and eventually they end up in what's known as a 6-helix bundle. And so at the post-fusion state, these 6-helix bundles have been fairly well characterized for other viral fusion proteins, and I believe there is a SARS-CoV-2 post-fusion structure deposited in the PDB. And so that's shown here in C on the illustration to the left. And so in between this, we don't really know exactly the specific steps. And that's mainly because these intermediate states of the protein are metastable and really hard to purify in a stable state and then characterized by high resolution structural biology methods. But overall, the S2 domain of the spike protein follows the trend of these class 1 type fusion systems, having a fusion peptide, some heptad repeat domains, and then an anchoring transmembrane domain. And so before we move on to thinking about some of the mutants and how this spike protein has evolved during the current pandemic, I just want to summarize by, again, emphasizing that the two main functions of the spike protein are, one, to be that initial point of attachment between the coronavirus and the ACE2 receptor in our host cells. And then after some cleavage events, then it performs its second function, which is to act as a fusion, a membrane fusion system, bringing the viral membrane together with the host cell membrane and releasing its RNP material. So we've seen this slide now a couple times, and so I won't belabor this point, but the summer IQB bootcamp students and summer RISE students did a lot of analysis on all of the spike protein sequences that were deposited as of late June. And so 1,190 unique sequence variants or USVs were identified in the spike protein, which is, the spike protein is only 1,273 residues long. So we're looking at almost one unique sequence variant per residue. These were found to primarily be double or mutants, 327 single mutants, and then a decent amount of multi-point amino acid substitutions were identified. These substitutions were located primarily in the boundary regions of the protein, with another large percentage at the surface. So there's lots of surface area and lots of residues exposed that can be mutated. And a very low percentage of these mutations occurred in the core. So these core, in this structure of the spike protein to the right here is a lot of the fusion protein machinery. So a lot of the S2 domain that's sequestered before the spike protein has been cleaved and it undergoes its second function. These mutations are generally non-conservative type substitutions. So, about 2/3 of these are non-conserved, found to be non-conserved. And then on average, it was found that, about 500 of the less stable mutants, these non-conservative mutations, resulted in an increase in Rosetta energy units of about 3, whereas stable mutations resulted in a decrease of about 2 REUs. So all of that is leading us to looking at the most common mutations that we have found in the spike protein. And by far the most common mutation is one that you've probably heard about, is this D614G. So as of the analysis of the GISAID data set, this count was up around over 20,000 instances out of about 40,000. So it's quite a dominant mutation. And it's mildly destabilizing, so it's not a hugely destabilizing mutation. And it's located in subdomain 2, so SD2. So that's the cyan domain here. And it's at the end of one of these beta sheets, so it's in a little beta turn region. And on the next slide, we'll zoom in on this a little bit and look at it in more detail. But it's not readily apparent from its placement in the SD2 domain why this has become such a prevalent mutant. And as we can see from the counts of the other most common mutations that were identified, they all include this D614G in addition to something else, right? So we'll ignore that for now, and we'll just take a look at where these other mutations tend to pop up. So the second most common mutation is D936Y, and it's a much lower count from the D614G, but around 200. And again, it's mildly destabilizing, even more mildly destabilizing than the D614 by itself. But this mutation pops up in the heptad repeat 1 domain, which is actually in this yellow helix here, which kind of faces the surface. So if I'm remembering correctly, this mutation is actually a surface-facing mutation, which is quite interesting. The next most common mutation is P1263L, and that is in the C-terminal or cytoplasmic tail of the protein, which so far has not been characterized at high resolution. So we don't have any information on how that affects the stability. Another mutation occurs near the fusion peptide region. And in this specific cryo-EM structure that was used for a lot of these energy calculations, that region is missing from the electron density, and so is not able to be modeled as well. And then the other two most common mutants that pop up are in the very end terminus of the protein. So this pinkish domain here is the NTD. And these are likely not showing up in the electron density map just purely because they're at the very end terminus and are probably quite dynamic and flexible. So if we zoom in on that most common mutation, D614G, and look at the difference between the aspartate and the glycine, we can see, or we don't necessarily see any kind of interactions that are perturbed right off the bat. So we're going from a larger side chain to essentially no side chain, right? But it's not quite apparent just from looking at the change in sequence why this is now found, why this is found in about 70% of all of the USVs that we identified and almost 75% of all of the SARS-CoV-2 genomes that are in the GISAID database. Work by others has identified in COVID-19 positive patients that this mutant is actually associated with an increased viral load, so more copies of the virus in these patients. But even more recently, Yurkovetsky and coworkers in this recent Cell publication actually found something that's a little bit more intriguing from a structural biology perspective. So what they did was they took a look at the D614, the wild type sequence of the spike protein, and then the D614G mutation. And they did at first some SPR analysis. And what they found is that they found a decrease in the binding affinity of the spike protein for ACE2 when you mutate from D to G. And this is a decrease of about 5-fold in the KD, although this is still fairly strong in the nanomolar, single nanomolar. But what they found when they looked at the mutant spike protein by cryo-EM and analyzed how many of the RBDs were either in a closed or non-binding versus an up or binding competent conformation, what they found was a very large shift in the equilibrium between closed. So in the case of the wild type, you have about 50-50 of the RBDs in a closed versus one up or one open position. But when you have this glycine mutation that pops up, essentially you remove, almost all of the closed variants of the spike protein, and 95% of the time, then you have at least one RBD in an up or a binding competent position. And so it's thought that this D614G mutation, even though it's in a location that's not directly next to the RBD, perturbs allosterically enough the ability of the RBD to pop into an up position. And so this just overall increases the availability of the RBD for binding to the ACE2, and therefore increases the amount of spike protein that can, or the amount of coronavirus that can attach to the ACE2 receptor. The other region I want to just kind of zoom in from a mutational perspective and take a look at is the receptor binding domain specifically on the spike protein. So the overall binding interface between the RBD and the ACE2, so the RBD here is in blue, the ACE2 is in gray, is very broad. And so there's quite a few different interactions that occur between the RBD and the ACE2. And what we found by looking at the different USVs that pop up, again, most of these include D614G, but if we think about specifically in the RBD, there's actually kind of a clustering of several of these mutants that are in this loop region here, kind of not directly interacting with the ACE2, but nearby the large binding interface. And so in our lab, specifically in the bomb lab, we're interested in kind of characterizing how those mutations affect this loop region and overall how that affects the RBD and its ability to bind the ACE2. Another region, it seems our hotspot of mutation seems to be these parallel anti-alpha helices here, although it's not quite sure, based off of this small construct of the RBD, how that might affect the overall stability of the RBD. So just to kind of wrap up and summarize here, D614G is now the dominant form of the spike protein currently circulating. And this is now kind of thought to be because it helps to promote the availability of the RBD to bind the ACE2. So it promotes more of these RBDs to be in an up and binding competent position. Most of the mutations of the spike protein are mildly destabilizing, but these mutations in this large protein pop up in almost every domain. And of course, as we move forward in our own research and other groups around the world move forward in their research, it is going to be critical to understand how the RBD binds the ACE2 in order to develop some therapeutics that will target this interaction and disrupt it. And with that, I'd like to acknowledge the students that were involved specifically in the spike protein work this summer, Erica, Sophia, and Lindsay. I'd like to thank my colleague, Cody Hoop, in the Baum Lab, who helped put some of these slides together and has been helping on the spike protein project in our own group. And I'd like to highlight the work of a very talented undergraduate, Andrew Sam, who I've been mentoring this summer, who has been helping me get a lot of these computational RBD simulations that we're doing in our group set up and running. And I'd like to thank Stephen, Christina, Sagar for putting together IQB Bootcamp and inviting me to talk today. Thank you.
Thank you, John. Let's see. We have a few questions already in the chat. Others, please write your questions in the chat if you have any. Antonio Meroli asks, the spike protein can have different angles with the membrane and it can bend, also thanks to its tapering stalk. Do we know if the protein breaks or can be cleaved at the passage between the intramembrane and extramembrane portion, or if it may be easily popped out from the membrane?
Yeah, so that is correct. It is definitely very flexible in the stalk, and a lot of the PDB structures that are deposited kind of ignore this because it's hard to characterize because it's partially membrane, transmembrane. As far as cleaving or popping out of the viral membrane, I'm not sure about that. I mean, there are no identified cleavage sites in the stalk region that I have encountered yet that would say that there's some enzyme that could cleave that off. of course, that could be a target for, a protease design as a potential therapeutic avenue, is to cleave off the spike protein from the coronavirus to begin with. But as far as I know, I don't know if any of those have been identified.
And John, it could be that there's an exogenous protease that could do the same job. I'm thinking about bromelain and its ability to cleave the influenza virus hemagglutinin.
Mm-hmm. Yep, correct.
Monica Roth asks, any information about the Mink adapted virus in spike and binding to ACE2 human?
Well, I'm glad you brought that up because I just actually read an article about that this morning. So let me just pull up a slide on that. So I prepared this morning because I read an article about this and I figured someone might ask a question about it. So the Danish Mink mutation is actually twofold. I believe there is a unique sequence variant that has 5 different mutants in it, as well as that has been identified to circulate in mink specifically. This mutation here, Y453F, has made the jump into humans, and I believe now in the GISAID database there are about 300 sequences that have made the transition to humans. And so what is interesting about this mutation is that it occurs kind of at this large binding interface between the RBD and ACE2. And so it's this tyrosine here that is mutated to a phenylalanine. So here, as you can see, this hydroxyl group points down to an associated histidine on the ACE2 at the binding interface. And there's a possibility for some sort of hydrogen bonding interaction to occur between these 2 residues. So if you mutate this away to a phenylalanine, you disrupt that hydrogen bonding, and you would hypothesize that this would be a destabilizing mutation. Now, how that affects the overall binding of the RBD to the ACE2, we'll have to leave it up to some experiments to be conducted, in the future. But knowing that the D614G mutation actually kind of also leads to a lower binding affinity between the spike and the ACE2, it could be that that after cleavage of the S1 domain, you need this a little bit lower binding affinity for the S2 domain to diffuse away. And maybe that is an avenue for why these mutations seem to be a little bit more advantageous for the virus. But of course, this will all have to be tested experimentally.
Okay, Hong Lee asks, what forces / amino acids drive spike trimerization? Could D614 be involved?
Yeah, so D614, let me see if I can pull up a nice, is on the outside of the protein, essentially. It's in this SD2 domain, which is the cyan colored region here with all these beta sheets. And so it's not necessarily at any of the binding or monomeric interfaces between, that make up the trimeric domain necessarily. So
actually, John, so if you look at that new paper, they have a cryo-EM structure which shows that there is, some backbone rearrangement, and D614 in fact makes a hydrogen bond with, I think, T6869 from another proteomer.
Oh, okay.
So it is involved in hydrogen bonds, and it sort of, yeah, exactly. She's right. When you disrupt it, it's sort of like, it acts like a latch, they think, to keep the RBDs down, although the RBDs are far away. But in any case, yeah, there are some interactions. I guess the sort of crazy thing to me about that cryo-EM study is that even though there is a significantly higher population of RBD up states, the binding is weakened. And I don't quite understand how that makes any sense. But
well,
do you have a...
As I kind of suggested with the Mink mutation, it may be that, well, if you look at the KD specifically, it's the on rate is the same between the E and the G, but the off is higher for the mutant, right?
Yeah.
So maybe it's this, after cleavage, it's important for the S1 domain to dissociate rapidly, allowing the rest of the spike protein to be accessible for its fusion activity possibility.
Yeah, that makes sense. Okay, let's move on to the next question. Can spike protein interact with the envelope or membrane proteins of SARS-CoV-2? If yes, what might be the role of such interactions?
I would think it's possible.
But yeah, I don't think much is known about that.
It's transmembrane spanning protein, so it's swimming around with those two other proteins.
Yeah.
But I don't think anyone's looked at that yet.
Yeah, that's a good point though. Ricky Cohen asks, when making lentivirus with spike for best processing while retaining similar binding affinity, would you substitute the VSVG signal sequences along with the transmembrane and internal domains? Would you recommend also mutating the furin site?
I'm not entirely sure how to answer that. So
Maybe if somebody, I know that Ricky had asked this question also on the mailing list. It seems if there are experts in this area, I think probably they should reach out to Ricky. Shishir asks, recent work by the Amaro lab has shown essential role of N-glycans at N165 and N234 site on the spike protein receptor domain conformation. I was wondering if there is any detailed analysis in your study or plans for future work on mutations identified for the N-linked glycosylation sites.
Yeah, so I have to go back and look in detail. I don't know if we've identified any mutations that happen at N-linked glycan sites. So I don't think these residues are mutated at all.
But yeah, I think that's a good point that we should look to see if there is any change there, because